A wonderful surprise to see this many people come here to San Antonio after some of the confusion and other things that have occurred not just in our nation but within this group itself. And we just cannot express our appreciation enough. Thank you very, very much for coming. This is the first meeting of the Americans, the American Pompe patient population. But of course, it's really a diverse group. You have people here from Australia, Brazil, Puerto Rico, Canada, other various parts of the world. So it's a very mixed group, and I hope you are able to meet each other and share in your experiences and trade some knowledge, trade some help, and look forward to the future as we all are. You know, my wife and I have been involved in this since our daughter was diagnosed about 6 years ago. There have been a lot of ups, a lot of downs, probably more downs than ups. But the good news is that there are 2 drugs on the market that work. They're not on the market, excuse me. There are 2 drugs in trials that we know work. And Marilyn and I know firsthand because our daughter is receiving one of the drugs, the drug in the Rotterdam trial. We're going to have 3 investigators begin their presentations this morning to explain some of the results and some of the experiences of the 3 trial centers. Dr. Arnold Reuser from the Erasmus University in Rotterdam has been instrumental in almost every aspect of this research. And he's going to give a general overview of the trials and some of those experiences. Dr. Ans Ploeg, also from the Netherlands, will discuss the Rotterdam trial. Andy Alpha-glucosidase, excuse me again, Andy, it's always hard to, but Dr. Chin's not here today, but Andy is going to give a presentation on some of the results and experiences of the Duke trial. And then we have Dr. Klinge from Essen, Germany, who's working with Dr. Voigt. They also have a trial using transgenic material and CHO material in Essen, Germany. I think he'll have some new experiences to share with us. The program is going to change, and we're going to have to just do it as we roll along here to see how much time it takes. The afternoon's going to change. Novozyme and Genzyme will want to address the audience towards the end of the day, and I'm sure you'll have a lot of questions for them because that's the next major event that's getting ready to take place. So just bear with us in the changes. We did have a larger room. Like I said earlier, I apologize for cramped space, but that's what a close family is all about. So I'd like to begin with introducing Dr. Arnold Reuser from Rotterdam, the Netherlands, head of genetics, Department of Genetics at Erasmus University. Arnold, please. Thank you.
Morning, everyone. There are still seats free over here. Maybe you can come forward. It's fantastic, I think, that there's such a crowd, because at one point we were thinking that we would not have this meeting, but since we're all here, I think the spirit is there, and I very much like the slip of the tongue of Randall House, that there are 2 products on the market, and you think, yes, they are on the market, but not quite so yet, but they will be on the market, and the question only is how fast can we get them on the market. It's still, I think, it's an enormous effort to get so far, technically as well as in business-wise decision-taking. I think if you join forces, and that's what we should do, should not quarrel about what's best. We should do the best, and if we do the best, I think we get it done all together.
Arnold, we're not sure if your lapel mic is on correctly.
This one better? Okay, I got it off on two sides, but now it starts to. Right, and I have to mention Marilyn House and Randall, the organizers of this meeting. They've been very instrumental in I would say, forcing therapy into existence. They have really taken initiatives that were not taken by anyone else. They have brought people together. Here in San Antonio, they did it now for 2 times in San Antonio. Actually, this is the first patient meeting. It's the 3rd one. And once we gathered in the lab of Paul Plotz and Nina Raven, who is here too in Washington, and it has been very, very stimulating. What I decided to do is give kind of an overview of an introductory talk about what's all about Pompe disease. To get you going and have a better idea and an introduction for the other lectures that are going to come. Okay, dim the light a bit. That's greetings from the Netherlands. These are my children 3 years ago, so they're still young, keeps me alive. This is Dr. Pompe. If you were wondering why this disease is called Pompe disease, it's because this Dutch pathologist who was born in 1901 and unfortunately died during the war in 1945, was the first to describe this disease. It was a very severely affected child with cardiac enlargement. So the disease was called after him Pompe disease. Although in the same year there was a German pathologist, Dr. Puchar, who also described this disease, but in German. And for whatever reason, the disease was called after the Dr. Pompe and not after Dr. Puchar. The next slide is immediately more complicated because Pompe disease, that's easy to understand, that to pronounce by everyone, but then if you talk about glycogen storage disease type 2, you immediately get into the difficult words. Glycogen is here in the middle and depicted here in this figure. Glycogen is a complex branched chain of sugars, and it serves in the body as energy storage. For instance, if we start, I don't have a pointer, but okay, if we start left, carbohydrate. What's carbohydrate? For instance, if you eat pasta or corn or bread, the food is digested in the stomach and in the intestine, and it's degraded then, the carbohydrate is turned into glucose, and glucose, it's a sugar, it gives you energy. So this energy has to be distributed to all the different organs. That is via the blood. Oh, I can do it this way. Okay, via the blood, goes to the brain, the liver, and the muscle. If you need that energy, you use it up and you burn your glucose. But if you don't need the energy immediately, the body stores the energy. And how the body does it, it links all these single moieties of glucose into this larger aggregate, which is called glycogen. And that's your energy reserve. When the energy is then needed for muscle function, sudden exercise, it is again turned into glucose, the single moieties. Oh, wonderful. There you go. Okay. Now there are 2 ways to convert the glycogen. There's the L pathway, which is lysosomal, and there is the C pathway, which is cytoplasmic. Now again, these are difficult words, cytoplasmic and lysosomal. The lysosomal for this meeting and for Pompe patients is the important pathway, because you have your muscle problems because you accumulate glycogen in lysosomes. And you accumulate this glycogen in your lysosomes because the glycogen cannot be degraded into glucose. And why not? Because one single product is missing. And this product is called alpha-glucosidase. So Pompe disease is a glycogen storage disease, and it's a lysosomal glycogen storage disease. In contrast to 9 other glycogen storage diseases, which have a cytoplasmic origin. You see how complicated it is? Immediately it becomes complicated. This is a cell. What is a cell? A cell is the smallest working unit, building block in your body. So a liver cell, if you have millions and millions of liver cells, you have a liver. If you have millions of muscle cells, you build a muscle. But this single muscle cell is still a muscle cell and able to perform all the functions that are required for contraction. So this is then the structure of a cell, not necessarily a muscle cell. And then what it shows actually, it's kind of a square block, but inside there are different compartments. The one in the middle, which is called nucleus, contains genetic information. And this genetic information is extremely important for the cell, because this genetic information tells how this cell needs to be built. It also tells exactly what type of processes have to go on inside this cell. And it also tells when these processes have to go on. So without this information, there's no cell, there's no function. This information, it's contained in the DNA, the genetic material, and that's the material that's inherited from parents to children. There are other compartments, difficult names again, a mitochondria, that's where you produce the energy. And then there is the lysosome. And the lysosome is a rather small compartment. And what's the function of the lysosome? The function of the lysosome is to recycle all types of material. The cell is not wasting anything. Everything is reused by taking it down into pieces and using the pieces again to build up a new cell. And that is how the cell stays alive and how we stay alive. So that's the lysosome. Now in Pompe disease, you have the glycogen, here and there, all these little dots. That's the energy reserve. And then this glycogen enters the lysosome to be degraded into glucose. But now, since the enzyme is missing, it cannot escape. So it accumulates inside this little organelle, and this little organelle is expanding, and there are more of these. So the whole cell is going to be filled with lysosomes, which are full of glycogen, and they disrupt cell function. This is a normal muscle cell. You see how fantastically it's built. These are thick filaments, as they are called, and here are thin filaments, and they slide like this. And if the muscle contracts, then the thin filaments slide in between the thick filaments, and the muscle becomes shorter. And when it relaxes, they slide out again. And that goes by the unit. So every unit from here to here continuously slides. That takes energy. Here's the energy. These are the mitochondria producing energy. And these dark grains, that's glycogen. Now in Pompe disease, you see that the structure already is different. This is not a very severely affected muscle. Here you can see these filaments. But now here in between the filaments, you have these what we call vesicles. That's the lysosomal compartment. And inside the lysosomal compartment now, here and there, you see that the glycogen is trapped. And that leads to muscle damage. So that's the mechanism of the disease. This is another way of staining, and you will see more of these slides. This is a muscle biopsy. This is how the tissue should look. This is normal muscle. And this muscle is from a very severely affected patient. And in between the muscle fibers, you see all these deposits of glycogen. As I said, the information to build the muscle and to make all the material that are needed in the cell is contained in the DNA. So the DNA is the genetic information on how and how much of any particular product to produce. Now for Pompe disease, it's important to produce alpha-glucosidase, this enzyme that degrades glycogen into glucose in the lysosome. So if there's any abnormality in the genetic information, we call that mutations or abnormalities, then of course immediately it results in abnormalities of alpha-glucosidase. These abnormalities can be quantitative, simply there's not enough, and they can be qualitative, it's there, but it doesn't function good enough. So for the diagnosis of Pompe disease, of course, there are the clinical symptoms, but also, of course, you can study, analyze the DNA, and see whether there are any mutations, abnormalities. If you find these abnormalities, you have a diagnosis for Pompe disease. You can also look at the enzyme, the alpha-glucosidase, and see whether there are any abnormalities in function. For instance, the activity, that is how much glycogen is converted per hour into glucose, that too can be measured. I forgot to mention one thing, because we have the acid maltase deficiency association. Now, acid maltase simply is a different word for alpha-glucosidase. Because if you have two sugar moieties, and if you link them, so you don't make this large aggregate of glycogen, but you simply link them one by one, it's called maltose. And also maltose cannot be split into glucose-glucose. So this disease is also called maltase deficiency. Okay. Thinking about therapy, there are two ways of therapy, and both will be addressed during this meeting. Gene therapy. And gene therapy is try to correct the abnormalities in the DNA or simply replace the DNA. So instead of having a non-functional or abnormal DNA, put a piece of proper genetic information in the nucleus, and that will tell how to make the alpha-glucosidase as it should be. The other way to interfere is enzyme therapy. If you don't have enough alpha-glucosidase or it doesn't function well, you simply replace it and feed the cell with more alpha-glucosidase so that it should take over the function. Now this is then the mechanism of enzyme replacement therapy. This is a very simple, this time a very simple cell with a nucleus. The red dots are lysosomes. Simply think about this little cell not sitting in the body but in a little dish, and you feed to the media and they will grow. The stars now is the enzyme, the alpha-glucosidase. The idea of enzyme therapy is that the enzyme will bind to the surface of this cell, and these blue beans, they may facilitate the binding. These are called receptors. So if now the alpha-glucosidase is bound to the surface, then this cell will internalize the alpha-glucosidase, and then it is transported to the lysosomes automatically. Nature will do the rest. So if you're able to feed enzyme to cells, you can correct the storage disease. Now in the body, it's far more difficult, because in the body, you feed the enzyme via the bloodstream, the blood vessels. But of course, the blood vessels have a wall. And first you have to cross the wall in order to reach the tissue in between the blood and the muscle. So here you have a pathway of diffusion where the enzyme should leave the bloodstream, cross the barrier, and only then binds to the blue beans, will be taken up by the muscle and internalized by lysosomes. So that is the challenge, to produce an enzyme that can do the job. And that's what's presently ongoing. How do you produce enzyme? Because you really need a lot. So you need a little factory to produce. Now at present there are two products. One being made in a Chinese hamster ovary cell. So again, this cell, basic unit of life. And here you have another cell, which in this case is a fertilized oocyte. Because also a fertilized oocyte is a cell. Except that this fertilized oocyte, of course, can develop into an embryo and then further develop into an animal. So what do? You take a fertilized oocyte or a CHO cell, you take the DNA, so the genetic code, which is dictating how to make alpha-glucosidase. So you take this genetic information and inject it into the nucleus, where the other genetic information is, of the CHO cells, or alternatively, you inject it into the nucleus of this fertilized oocyte. If you take these cells, put them in a reactor, stir them, they will start producing alpha-glucosidase, and you can harvest the alpha-glucosidase either from the media or the cells. And if you produce, for instance, a rabbit, as was done by farming Enzyme LLC, you can collect milk from the rabbit, because these cells, they make, I mean, you find them back all over in the body, this fertilized oocyte. So you can then collect the milk from the rabbit, and from the milk extract the alpha-glucosidase. This product now is being given to patients in the two trials you will hear the result of. Okay, this is a kind of a second theme in my presentation. It's about what we are doing in Rotterdam. We call it the Pompe team Rotterdam because it sounds nice like PTR, Pompe team Rotterdam. The activities, fundamental. Okay, what's fundamental? I will read it for you because it's really bad. Okay, we study structure and function of alpha-glucosidase in order to learn about this enzyme. We study structure and function of the gene in order to learn about gene function. We study correlation of genotype and phenotype. That simply means try find the mutation and see what it does to the patient, so that by looking at the DNA we can give a diagnostic answer or a prognostic answer. Then of course we have studied how to produce alpha-glucosidase, and also we are studying how to apply and evaluate enzyme replacement therapy. This is the applied side. Of course we offer diagnosis at the DNA and protein level, and also try to give prognostic answers by knowing the natural cause and by making the genotype-phenotype correlation. And then of course there's the clinical study you'll hear about later. One very important thing in this, we get this flood of information and we kind of try to challenge that, put it in a format so that it's accessible. And the format is the database that we started to set up 2 years ago and that we are now filling. This database has all kinds of forms with information on, for instance, the family, so clinical data, and also pedigrees. It has forms about, with all publications about Pompe disease. We have information on mutations, etc. Now the list of mutations is most complete. I can show it. The mutations in the DNA are listed here simply by the number. The location is listed, so we know exactly where they are. We know exactly what the effect of each mutation is. And we also know what it does to the alpha-glucosidase. This column lists whether yes or no the mutation has been published, because many of them have not yet been published. And some extra information where the material came from. For instance, what is it here? Pakistan, Germany, Australia, Japan, Israel, and France, and of course Dutch. So we keep track of all this information. And then also for each information we try to we try to have as much information as possible about what exactly it does. For instance, here we have a mutation, it's called T307G, it really doesn't matter. We know what it does on the amino acids, so on the alpha-glucosidase. We know whether it's severe or whether it's mild, and this mutation was discovered in a patient from Australia. Then at this side it says how much activity there is left if you have this mutation. Now the normal value is up in the 2500, and this mutation doesn't have any residual activity. And then also we study what it does, in this case, to the synthesis of alpha-glucosidase. Now this is normal. All these black bands simply tell that these cells produce a lot of alpha-glucosidase, and the patient does not produce any. Then if you would click a button here, the button Materials, you get another tab, and then it gives a list. In this case, it's a different mutation. What kind of material contains this mutation? And for this mutation, it's a mutation that's very common in the Netherlands. We have a list of 42 patients with this mutation. And the database tells exactly what material is available, and by clicking further, where we have stored. This is the information on the patients. It's all anonymous, so they have a unique identifier number. The form tells you that this patient had the adult form of Pompe disease. The year it was diagnosed, 75 in this case, a male from the Netherlands, and the date of birth. On this tab, we have the family relation, so the relatives. Now this is a very particular situation, because this is the patient. He was diagnosed with an adult, late onset form of Pompe disease, but 3 of the 4 grandchildren had severely infantile form of Pompe disease. So also we keep track of that type of information. Of course you want to know who has sent in this family for diagnosis. We have a contact tab, and it will immediately list the name of the physician or the institution, and we will know where the cell line came from. Then we have the material. Oh no, this is the diagnosis, so it tells you what type of assay was used to diagnose these patients. And this is very important, because every laboratory still has its own procedure, and data are not really comparable between laboratories. So you always need to know which laboratory, you need to know standard ranges, etc. And then the last form, and also the last slide then, that's the material form again, and it will tell for each individual patient whether yes or no we have sequenced the gene completely, or whether we have only screened for common mutations, and what type of mutations we found in this particular family. So this is very short overview of the database, and kind of indicates what type of work we are doing and where we think is important to concentrate on. Thank you. If there are questions, I don't know how we're doing with time.
Yes, we're fine.
Yes?
If a patient has some enzymatic activity, is it prudent then to limit the intake of carbohydrates? Because what carbohydrate and glucose is ingested, you burn that up within a short period of time. Is this an effective in slowing down the progression of?
I'll repeat the question. So the question is, what can you do in terms of regulating your diet? There's not so much known actually about the effect of diet, but it's advised not to take too much carbohydrate and use a high protein diet, because if you take a protein diet, at the end, protein means amino acids, But inside the body, the amino acids are again converted into glucose. So it may help a bit, but it doesn't really, I don't think it has a very large effect. But it is advised to have a low carbohydrate, high protein diet. Ans, you want to?
When you are in a catabolic state, that means when you don't eat enough energy, then you break down proteins. So you also break down the proteins of the muscles. So it's always important to have sufficient energy intake and sufficient protein intake. I think that's important. Yeah?
Yeah, so I think so, yeah. And then exercise is another thing, because if you exercise, you burn your glycogen. But if you exercise too much, it may be too strenuous and you damage your muscles. That's for the physiotherapist. That's very little hard data, just to tell exactly you should do this or you should not do. And what we notice that patients are experimenting and doctors are experimenting because no one knows exactly. But I would say if you keep it a bit low on carbohydrate and a bit high on protein, and if you exercise a bit, it's probably best. Agree? Other people that want to agree or disagree, but okay.
I've got a question for you. Is there a central database, worldwide, or is every group, like you have your significant database there that you registered the mutations with? Is there a central group that all?
There's not yet a central database. And we have started to set this up 2 years ago, and we are still filling it. And the idea is to publish parts of it at a regular basis. So, for instance, for the mutations, to have it on the internet and then to update it regularly. Of course, patient information we cannot distribute all over the globe. So that's more of a difficulty. If someone would call me and ask me, can I have this or that type of information? Yes, it will be, of course, free and for everyone available. Yeah.
Well, do certain mutations burn a certain percentage of the glycogen? Does it break down? Can it have a small amount of the enzyme versus a normal?
Okay, there are some mutations you definitely find in one group of patients and definitely not in the other. So by looking at mutations, you can say this is late onset Pompe disease and it's definitely not the very severe infantile form. Other mutations, just by looking at the mutations, it's hard to predict. You have to do further investigations. Yeah.
Got one more question. So you can differentiate the infantile, the adult onset, which comes later in life, usually doesn't affect the cardiac. It's usually a pulmonary aspect of it first, but
right.
Initially it's a rapid onset with the juvenile and infantile, but so that must be a different type of progression or the genetic aspect of it.
Patients with a milder form of disease, they usually have residual activity. So that means the gene is not damaged to such extent that there's no activity left over. So you do still convert small amounts of glycogen. And the rate of accumulation is slower and the rate of muscle damage is slower. And of course you have muscle damage, but you also have muscle repair. So it's continuously a competition between damage and repair. If you have some activity left over, if you don't have any activity left over, it's only damage and there's nothing to repair. But those are the infants and not the late onset. And since there still is repair, training, some exercise may help, because by exercising you build muscle. I mean, the bodybuilder, that's simply exercise and building muscle. So you are able to build muscle if you do it the right way.
Got one more question. If you're looking at a patient that's, actually has, diagnosed with Pompe's, and you're looking at trying to burn it, you look almost like a modified Atkins diet. Is that pretty severe for somebody that, say, you had a patient on a ventilator, and you want to, help them lose a little bit of weight at the same time you want them to metabolize? Do you understand what I'm saying?
No, I didn't get a question.
Well, I'm confused anyway, so that's good. Thanks.
Do I introduce the next speaker, or do you, or the next speaker is just coming up?
We're going to make an adjustment on the projection.
She has PowerPoint, so I think that.
Okay. Is it okay like this? Yeah? Should I start, or is somebody bringing up a little table for my computer? Nee, iets dichter mee. Oh. Yeah. Is everybody ready? Can I start? Yes? Okay. I would like to present my data, or our data, on the trial in Rotterdam. We perform an enzyme therapy trial in Pompe disease with recombinant human alpha-glucosidase from rabbit milk. We started this trial about 2.5 years ago. And first of all, I appreciate it very much that I can be here today and present the data to you. I said it was the first clinical test in patients, and the trial was supported both by farming and Enzyme. And as you know, there are different variants of Pompe disease. There's the severe infantile form, which is best defined, and then there are the late onset forms of Pompe disease, who are also subdivided in childhood, juvenile, and adult form, but I think it's more a spectrum of diseases. First, I would like to give you a short introduction to the natural cause of infantile Pompe disease, because when you want to interpret the data, you have to know something about how the disease evaluates. Alpha-glucosidase is the protein that is deficient in Pompe disease. And Arnold already mentioned the level of alpha-glucosidase determines the severity of the disease. In infants, alpha-glucosidase is almost absent, while in late onset patients there is a residual amount, and activity of alpha-glucosidase. This is a baby with Pompe disease. And this is how it classically presents at the end stages of the disease. And you see how fast the process goes in the baby. Of course, there is a variation in this disease. And first of all, we did a study, and we studied 20 patients with infantile Pompe disease, which were diagnosed in Rotterdam. And we compared those data with data obtained from the literature. With this knowledge, we started the first clinical test in infantile Pompe disease, or in Pompe disease. In total, we started with 4 babies with Pompe disease, and later we also included 3 late onset patients. First, I'm going to talk about the babies. The trial started in January 1999, and we started with the product produced in rabbits. Arnold already explained how it is performed. A piece of extra material of human alpha-glucosidase DNA is injected in an oocyte. The oocyte is, the fertilized egg is placed in the foster mother, and then a rabbit is born who makes, finally, alpha-glucosidase. The rabbit is milked, and here, from the milk, alpha-glucosidase is purified and finally supplied here via a porticate system that is to make it easy for the baby, and put or given, administered intravenously to the patient. In total, we included 4 patients in the trial. Here you see the age of onset of the disease in the patients. They had all onset of symptoms before 3 months of age. The age of diagnosis, they were all diagnosed before the age of 6 months. And here you see the age of inclusion. And what is important here is that we included 2 patients rather early, when they were in a good clinical condition, and 2 patients who were in an end stage of the disease. And one of the patients you have seen before. And if you can remember the mean age of survival of the babies, then these babies were rather old and had severe symptoms. I think that's important. They all had a cardiomegaly. The younger ones didn't need oxygen, and the older ones did need oxygen. One patient became ventilator dependent at start of the trial, and the other one 10 weeks after start of the trial. We looked at the alpha-glucosidase activity because we wanted to be sure that all these patients had the infantile form of Pompe disease. And here you see the activity in fibroblasts and in a skeletal muscle biopsy. And you see that the alpha-glucosidase activity is very severely deficient if you compare it with the control range. 42 to 160 in fibroblasts, while the activity in the patients ranges from 0.1 to 0.7, and here 8 to 40 for the muscle, while it ranges from 0.2 to 0.4. You can look at the activity, but you can also look at protein. This is how protein is produced in a normal patient. You see normally 3 bands. And this form of alpha-glucosidase is the active form. So that's essential for the enzyme to work. And you see when you look at the patients, you see that 3 of the patients do not make any protein and 1 makes a tiny little bit. But we also learned that it matters what kind of diagnostic tool you use to see whether they make a little protein. Because with this method, you see that 3 of the patients produce a little alpha-glucosidase and only 1 doesn't. By looking in particular to the defect in the alpha-glucosidase genetic code, these are the defects, we could learn that the combination of this defect indeed takes care for not producing any protein of alpha-glucosidase. Is that clear? Because this is a little complicated. If there are any questions, please let me know. Of course, most of the information about the genetics and whether they've produced protein and so on was done during the trial. And now I go to the trial. The enzyme was generally well tolerated by the patients. As you can see here, this is one of the patients on his bike, and here he has the alpha-glucosidase infusion while he's playing. You see a lot of figures over here, but this slide shows the alpha-glucosidase activity in the muscle. When we started, all patients had a severe deficiency of alpha-glucosidase, I already showed you. And we started with a dose of 15 to 20 milligrams per kilogram, and then after 12 weeks, we took a new biopsy. And we were happy to see that there was a significant increase in the activity. The activity increased to 10 to 20-fold the onset value, but you also can see that it still did not reach a control level, because that's 8 to 40. Because also the patients were very severely affected, we decided in combination with these results that we were on the right track, but it was good to increase the dose, and we increased the dose to 40 milligrams per kilogram, and 12 weeks later we took a new biopsy, and here you see that all patients now had a normal alpha-glucosidase activity. And we again took a biopsy at 72 weeks of treatment, and then again, more or less, the activities remained the same. The enzyme we gave was a large enzyme. It's maybe not so important. But if you look here in the patient, this is a slide from a patient, and you see no bands of alpha-glucosidase. That was before treatment. Then we supplied the alpha-glucosidase, administered it to the patient intravenously, and this is 72 weeks after start of the trial. This is what we supplied. You see it over here, but you see also that you see now the lower bands. And these lower bands reside in the lysosomes. That are the active forms that are needed to do the work. And for comparison, I show you here a healthy fibroblast of a healthy individual. And you see that the same molecular forms are found. So this is very important. You give this form, and the patient doesn't have it, and then from the muscle, we retrieve the active alpha-glucosidase. So that's very important. This is what happened to the glycogen levels. And I just want to show you is that when patients, that in the best performing patients, there was a significant decrease of the glycogen level in the muscle, but that was more or less remained stable in the other patients. We also learned that it is very important how the disease situation of the patient is at start. The most severely affected patients showed very much damage. And if you can remember the slide of Arnold, where you have the structure of the muscle, it's hardly not seen anymore. It's gone. So it's very much destroyed. The slide is stained with a certain dye so that you particularly see the glycogen. So this purple is all glycogen. So we gave alpha-glucosidase, and here a biopsy after 72 weeks of treatment. And you see in this patient, and this is one of the late treated patients, that there is still a lot of damage. There are also good fibers here in between, but there are also many fibers who still have damage. You see that the purple stain is less, but the muscle did not completely restore. And this is what we see in the best performing patients. Very much damage also at start, but then 72 weeks after treatment, it's almost normal. The first effect we saw on the heart. I showed you earlier that before start of treatment, the thickness of the chamber of the heart increases with age. And that's what you see here in all the patients in the pre-treatment phase. But then after start of treatment, this changed, and the thickness of the heart became less. And here, in these two patients, when it approaches the upper limit of normal, it levels out. We also started for the mass of the heart. If you look at the mass of the heart in grams per square meter body surface, if you look at the grams of the heart, you also see the same. They were all enlarged at start of treatment. This is the upper limit of normal. It's about 100. And then you see it decreases under treatment. And again, in these two patients, it levels out when it approaches the upper limit of normal. At start of the trial, we were very much concerned about the mental development of the children. Because we know that in the infants, there's also glycogen stored in the brain. And we know when you treat those patients with alpha-glucosidase, the alpha-glucosidase cannot pass the blood-brain barrier, so it cannot enter the brain. But happily enough, we saw that all patients showed an improvement in the mental score. And here I show you a videotape of one of the late included patients. And here the mother asks whether she can give her the round with the proper dye. So she asks for red, for blue, for green, for yellow. And each time she makes the right choice. But you can also see that when we start treatment late in the patients, there's also residual disease. And we learned that it is very important to start treatment early. This is a very young patient at start, 3 months, and she learned to sit. And here, another patient, you see that he is not able to lift up his head. We call it a head lag. And here, the patient at 84 weeks of treatment. And this is a videotape of the patient made very recently during an alpha-glucosidase infusion. Oh, I'm sorry. Okay. What we learned is that the treatment is effective, but we always also saw that there are response differences. And what explains the response differences? Is it the uptake in the muscle? I say no, because all patients showed a complete correction of the alpha-glucosidase activity in the muscle. Is it the type of mutations? No, because all patients had mutations that are responsible for the severe infantile form of Pompe disease. Is it antibody response? We don't think so, because we saw the highest response in one of our juvenile patients, and I showed you one of the patients that learned to sit is one of the patients that does not make any alpha-glucosidase protein. What I think that is the explanation for the differences of the response is the state of the disease at start, so it's important to start treatment early, and the age of the disease at start, and those two can be related, or mostly are related. So my recommendations for the future are start treatment as early as possible, and also provide multidisciplinary care, because it's important to give the patients physical therapy and to have them as mobile as possible. And what is happening with the late onset juvenile patients, I will tell you later this day. Thank you very much.
Hello. What type of multidisciplinary treatments did you include during the clinical trials for the children, specifically?
Now, at least every patient receives physical therapy, and that's mostly that was installed before start of the treatment, and in fact in all cases. And if needed, if the patients have residual problems like contractures or respiratory needs, we add the home ventilation team and a good rehabilitation program.
Yeah, the 72-week levels, the 72-week enzyme levels, I think it looked particularly like patient 4 showed a decrease.
Yeah.
Are you able to say why that was and if it's clinically significant?
Yeah. You noted it very well. I tried to show you all the data. I do not really think, but then you should follow up, that it is really a significant difference. What we learned is that it can change per piece of muscle you take what the activity is. There is some variation. And there was also a patient who, for example, was earlier, I thought, I do not have it completely in my head, but that they first started to be 13, and then later, in the later biopsy, it was 29. But if you look at the range, it's more or less, in both cases, in the same range. Yeah?
You explained that it's important to start the treatment as early as possible. As time passes and the disease ages, is there a time in which that the treatment will become less effective?
Yes. Of course, I have to state that there are still limited results, but of course, we had a very thorough follow-up on the patients. And what I learned from the treatment is that it is an effective treatment, but most effective when you start early. And what I also learned is that infantile Pompe disease is a very devastating disorder, and that it is very important to have an effect of treatment, that there is still some activity of the muscles, that the muscles still act. If there is no action of the muscles, it's very difficult to get them started again. That's something, yeah.
I think I can talk loud enough. The muscle biopsies you did, is it showing equivalent levels of improvement in all muscle types, or were there different ones in different types?
Of course, when you take a muscle biopsy, it is an invasive procedure. We always took a muscle biopsy, an open muscle biopsy, so it's a surgical procedure. And why did we do that? Because we wanted to be sure that it was muscle that you take and not fibroblasts or fat tissue or so on. We always took it from the same spot, from the musculus quadriceps. 40% of the body consists of muscle, and you have many muscles. So it's simply impossible to take a part of all the muscles. But what I learned is that what you see in the patient and what you see happening, how the muscles act, that very good resembles the condition of the muscle. Is that an answer to your question? So all muscle biopsies were taken from the musculus quadriceps, and from that I've learned that what you see under the microscope very much resembles what you see in the patient.
Just from your response to my question, are you saying that in order for it to be effective, if a person receives the treatment at a later age, there has to be at least some muscle activity or some muscle movement?
It's a very difficult question you ask me, and it's also a very difficult discussion, and maybe we should talk about it a little later in person, and we can go in a little bit more detail. But as you have very good and very hopeful new treatments, and happily enough, there are also limits to treatment. And that's something we have to talk about with physicians very, 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 very good. We have to open that discussion.
I had a couple of questions. First of all, does it improve lung function?
Who was asking the question?
It's over here.
Okay. I will show something. I will also talk about for the infants. You mean?
For adults.
Yeah, but I'm going to talk about it later.
Okay. And also, and this is probably jumping again too, but just seeing the incredible differences in the infants. How are the individuals? I've met some incredible people just in this room. Who decides, and is this going to be available for everyone, for all of the AMD patients? I mean, and how soon is that?
Yeah. Now, that's also a question that will be addressed later today, and that's one of the reasons why you're here, also to hear more about that. Of course, the main target of doing a pilot trial as a doctor is having the treatment available as soon as possible for everybody here in the audience, and not only for a subset of patients. And I think that's the target. But of course, you have to have the material, you have to have the enzyme to treat the patients, and that, of course, is my limit. And then you are dependent on the industry to provide the material.
Yes, my question is over here.
Yes, I think you make a very good remark. Is it not important to start the next presentation? Because and then we have some questions at the end, because I
That's
think
fine.
very much is about the same type of, and maybe then you have even more questions and we continue.
Go ahead and plug me in. While we're getting this set up, my name is Andrea Amalfitano. I work at Duke University Medical Center. I'm a pediatric geneticist. So that means I've been trained in pediatrics as a clinician. Actually, some of my trainers when I was at Mayo Clinic are here, and I was pleased to see Dr. House here. He's one of my mentors, and I really learned a lot from him in learning to talk with patients and trying to educate patients. Because I view what I've done in getting a Ph.D. to do research as well as to learn to be a doctor is I need to translate this information that's so complicated in a way that you can understand it so that you can make intelligent decisions about what's happening in your lives. And I do this not only for Pompe disease, I do it for every genetic disease that I encounter in every day. And so that's what I'm going to try and do today. Some of it's going to be a little reiteration. Some of it's going to be a little difficult for me because my ears are still plugged from the airplane ride last night. So you may have to speak up a little bit. But I want to tell you a little bit about what we're doing at Duke. And the first thing I want to say is that at Duke, the primary emphasis has been how do we get the enzyme to the most people in the quickest amount of time. And I will tell you that that drives every decision that we have ever made. And I want to show you how we progress through that. There's two talks, actually. Dr. Chen is supposed to give this talk, and I'm not worthy to be in Dr. Chen's shoes. He wasn't able to be here, though. Some of it had to do with the confusion. Was the meeting on? Was it off? But I have been involved in the clinical trials going on in Duke, and I'm going to present that enzyme data. As well, my laboratory is very much interested in gene therapy approaches for not only Pompe disease, for a number of other diseases. I'm going to hold that part of this talk until tomorrow because we want to make room for some of the pharmaceutical company reps here. I know you want to hear from them. You don't want to hear from me. You want to hear from them. And I will move that talk till tomorrow. Okay, so we just have to hang on and listen for that. Now, Duke, we're known for basketball, and I feel that we're taking that into our research realm here. We're applying a full court press to Pompe disease. We're looking at every angle. We're looking at enzyme therapy, gene therapy. We're turning over every stone, trying to come up with ways to treat this condition. I'm going to focus on enzyme therapy this morning. You've heard there's three clinical forms: the infantile form, the juvenile, and the adult onset. And I don't have to tell you what these diseases mean. You guys are living it. You educate me, actually, when I meet with you, and you tell me about your diseases. You know more about it than I do. They're all due to this deficiency of this enzyme called acid alpha-glucosidase, or GAA, or whatever you want to call it. We call it GAA at Duke. What happens? You're missing this information, this genetic information that codes for this protein that breaks down glycogen in your muscle. So if you're missing that information, you accumulate glycogen in the muscle and you get your symptoms, okay? It seems to be that it all boils down to this. The DNA you inherit, and you know this is a genetic disease, it's inherited, encodes the information to make a protein. So if you have a mutation in the DNA, then you don't make the protein. And what Dr. Reusser and Ans were trying to explain earlier, which I think they did a great job, is that depending on the mutation, you either produce a little bit of protein that doesn't work so well, and that's enough to get you through to maybe adulthood. If you're infantile form, it means you make absolutely no protein, and your glycogen accumulates much faster, and you present as a newborn patient, okay? So there's all this variability, this gray area. Depending on what mutations you inherit, depends on and what happens with the protein will give you your symptoms. And if you lack protein, you have the condition. Now, Pompe disease, as we heard, it's a metabolic myopathy, lack of this enzyme. This is a typical infant. Many of you have lived this. I don't have to show this slide. Baby is born, looks normal. Within the first few weeks of life, has repeated respiratory tract infections. You go to the doctor says, hey, he's got a cold, give him this, give him that. But it happens over and over. You keep telling the doctor something not right. And finally, the doc gets a chest x-ray because this kid's always getting pneumonias. And you look, and the doctor now realizes something substantial is going on because here's the chest x-ray. And in the chest, there should be a lung over here. But instead, in this child's chest, it's filled with this massive heart. Okay? So the doctor starts looking at the baby and says, you know, the heart's enlarged, it's a muscle. And look, this baby doesn't move around very much. Other muscles are affected too. You look at that, and now the doctor says there's a muscle problem going on. You get a biopsy, and you look under the microscope, and as Dr. Roycer showed you earlier, here's normal muscle fibers, and there's these big lakes of glycogen. You can't miss this under the microscope, and this is the problem. Okay? And if you look, these fibers are like cables. They're on a suspension bridge, and they're actually being pushed aside because there's so much glycogen in there. That's why that heart is so big. It's stuffed so full of glycogen. And eventually gets to the point where that muscle can't contract the way it's supposed to. When it loses that function, that's when you have symptoms. So if the heart can't pump effectively, you end up in cardiac failure. And that's what, unfortunately, all the infantile patients eventually succumb from. Now, the juvenile and adult onset patients, it looks like you inherit mutations that allow you to have enough enzyme to sort of let the heart muscle survive, but your skeletal muscle is involved. And depending on how much enzyme you have left will determine how severe your symptoms will be and when they will come on. And that's where it gets a little more variable, too, even within families. I'm not going to go through this because I think our previous two speakers described just the infantile Pompe disease. Clearly, if you have no enzyme activity, you will have heart involvement, and your prognosis is not very good. Typically at Duke, if you're diagnosed at the age of 5 months as an average, the disease, we only have 3.5 months before we watch babies start dying. Adult onset patients, they can present all kinds of ways. And I'm sure you all have your stories of how you present it. Typically, it turns out a lot of people are showing up in emergency rooms in respiratory failure because they have a cold or a pneumonia and they end up on a ventilator. And no one can explain why. And then you get a history of, oh, you know, the last couple of years it's been harder for me to walk upstairs and I can't get out of my beach chair the way I used to, and then you turn out and have this diagnosis. And we're diagnosing a lot of adult onset patients at Duke because we've trained all our pulmonologists to say, if you see someone come in with an unusual respiratory failure, diagnose this because it's out there. It's actually more frequent than probably we all realize because no one really knew to look for it. Now, the key to treating this condition, believe it or not, Pompe disease, there is an advantage to having Pompe disease than having other muscle diseases. And it's all because of basic biology that Dr. Roycer showed you. Your DNA encodes for this enzyme called GAA. And that produces this protein, this wonderful enzyme that you're hearing about. This enzyme has two things it has to do. It has to find muscle cells, and it has to find its way into lysosomes to break down glycogen. Okay? So it's got two activities. There's a little part of it that signals where it's supposed to go. Then the other part breaks the glycogen down once it gets there. Both those activities have to work. One works and not the other, it doesn't work. And so there's receptors, as we heard about, that are inside the cell that grab hold of that enzyme and drag it into the lysosome where the glycogen is waiting to break down. Okay? So this is the cycle. Now, there's a quirk. There's a little trick. There's a little thing here that has allowed us to be taken advantage of. Some of this enzyme actually can get secreted out of the cell. Okay? And if you have a second cell next to this cell that can't make GAA on its own, and you put some of that enzyme on this cell, this second cell, it turns out that those receptors actually hang out on the outside of the cell for some reason. They're out there waiting. And they grab hold of that enzyme just the same way they would inside the cell. And they grab it, and they drag it into the lysosome. And that second cell can now have its glycogen reduced, even though it doesn't encode any of that information. Okay? So this is called cross-correction, and this is what we're taking advantage of here. It's just simple biology that we're kind of warping to try and treat this condition. So how do you treat a genetic disease like Pompe? There's two ways. One way is to correct the DNA, which I'm going to tell you about tomorrow, how we think we can maybe do this. Second is, okay, well, replace the protein. If that's what's missing, replace it. So this is what we hypothesized at Duke a long time ago. This is Dr. Chen's work, Dr. Van Hove's work, who's here as well. If you could make enough of that enzyme that I showed you earlier that retains those signals for being targeted and is still able to break down glycogen, if you could inject it into the bloodstream of an individual and let the blood do the work of delivering the enzyme to all the muscle cells, and those muscle cells, if they have the receptor still on there like we hope, they can grab hold of that enzyme and then drag it into the lysosome to break down glycogen. This is the theory. This is what enzyme replacement's all based on, this biology. Okay? So everything has to work. You have to make the enzyme. It has to retain the signal. It has to be made in such a way that it retains these signals so that that receptor can find it, drag it in. And then once it gets in there, there's all kinds of steps to be processed in order for it to work effectively. So some of the progression at Duke, the gene for GA was cloned. As you heard, there's a cell line, a CHO cell line, these Chinese hamster ovary cells that just love to turn out this enzyme in mass quantities, and we're trying to grow them up as fast as possible at Duke as well as at Genzyme. The progression, 1995, 1996, was when we first had enough enzyme to treat some animals, so we could go to FDA and say, look, we got some results in animals, let's try humans. And in 1997, Duke obtained this FDA orphan drug designation for Pompe therapy. What this is allows companies who are interested in this to know that they could potentially protect by patent their product if they choose to invest the amounts of money they need in order to make a lot of this stuff. Okay? So the enzyme, actually, Dr. Chen in his lab made enough enzyme that we were actually able to treat a bird. This is a quail. And I'll give you one guess which quail has the disease. It's this one here that can't get up. Okay? This quail has the same condition as humans with Pompe disease. So they have muscle weakness. You put them on their back, they can't flip over. You hang them upside down, they can't peck your fingers away like a normal bird can. They can't fly. And so Dr. Chen made enough enzyme and injected into these birds to show that you could correct the glycogen in the muscles of these birds, and that prompted us to move ahead into the clinical trial. Okay? So animal studies are very important, and we're going to continue to use animals to test everything before we risk humans in this. Now, I want to make this disclosure. I think it's important, and Duke wants me to make this disclosure, and I agree with it, that since this was invented at Duke, a lot of this, we have to make very clear that if anything came to be a product and somebody made money, there is the potential that someone at Duke could make money as well. Right? And so I don't feel too bad about showing you this because this is just the way it is. This is the way universities protect their intellectual property. But not only that, it allows a company to come in and know if we make a product, we're not going to get undercut by somebody as soon as we spend $80 million to develop this enzyme, and then somebody just learns everything we did and then undercuts us. You have to have that protection. Now, we published, now I'm going to go into our clinical trial. This is the paper that we published describing our results using this CHO-derived enzyme. It's published data, and everything I'm going to present is in graphs in that chart, in that table, so you can, in that paper, so you can go and look at it yourself. As you can see, these are all the authors. This is me up front here. A lot of people are, Andrea, they see my name, they're expecting, where the heck is she? Where's this Andrea at? And unfortunately, that's me. I have only one X chromosome. I have a Y chromosome, unfortunately, but I still seem to muddle through. Dr. Chen is the lead author on this paper because he spearheaded a lot of this trial. And everybody else involved here, and there's more people involved as well, helped us move forward to test this. Now, we have 3 babies enrolled. They all have infantile Pompe disease. It's been confirmed by DNA testing and by enzyme testing. We've shown in our paper that even these families suffered losses from having previous children born with infantile Pompe disease. So there's proof that they all have infantile. And here's the x-ray I showed you earlier. Here's one of the patients we treated before getting enzyme and 12 weeks after getting enzyme. You can see that the heart shrinks in size. In fact, all the patients showed the same thing, before and after, before and after. And x-rays aren't really good. You can look at a picture and say, yeah, that heart looks smaller. But we actually measured by echocardiogram, just as Ans showed you earlier, and showed in that paper that two of the three patients had dramatic drops in their heart size as measured by mass over time. And the third patient didn't have much to begin with, but certainly didn't progress as his brother did. Now, this is why I was asking on some questions, because this is where it gets murky. Okay? This graph here, and I want to show you this hard data because I think it's important for you to understand. We have ways of measuring muscle strength. It's called an AMDA test. So you take a baby, you ask, can it sit up, can it not sit up? Can it raise its arms, can it not raise arms? And you give it a score. Score of 4, 5, as they get older, their score should go up. The range of normal is within these dotted bars, 5 percentile to 95th percentile. Sort of like a growth chart. You've all seen them for your kids. This is the normal range for people that we measure. So when you're 4 months old, your score should be somewhere within here. When you're 8 months old, your score should be somewhere in here. Here's the results we saw in this trial. All patients, when you first look initially when they get enzyme, and that's the arrow here, initially showed increases in their AIM scores as we measured it by this test. 1, 2, and 3 certainly. Okay? But look what happened with time. Patient 3 continues to be in the normal range. But we had a divergence here. 2 of our patients, though they initially showed this increased muscle strength, eventually went down. Okay? And they're both now on ventilators. So sort of, this is why I was asking Ans to clarify, because we're seeing the same thing. Some of our patients, we have a dramatic response. Other patients, and all patients, their heart's correct. Okay? So we've converted infantile patients to, at least in 2 out of 3, to a severe juvenile form. We've corrected their heart. That's why we say that this enzyme works, because we know their heart responds. The skeletal muscle, that's where it's a little bit more divergent, a little more confusing. And I think this is why Ans is hedging on answering some of your questions, sir, because we're trying to figure out what's causing this. Okay? Now, Duke, we have one idea, but it's not the only idea, and we're just trying to throw it out there and trying to investigate this in our future trials. So we've got this word here, CRIM, and you heard our previous speakers talk about it. I want to explain this a little bit. So Western blot, so you take protein out of a muscle or skin out of a patient. A normal person, if you look for GAA enzyme, you see this black blob there. That's all you need to know, okay? In our 2 patients that initially showed responses, but then, and their hearts are still corrected, they eventually declined their muscle strength. They also did not produce any detectable enzyme by this assay. But our 3rd patient, we were able to detect some of this. Okay? This also correlated with production of antibodies. You know, what are antibodies? They're a way of your body detecting whether something has come into you and is foreign, right? Patient number 3 did not make antibodies by our detection methods. Patient 1 and 2 did. And when they made antibodies is when their muscle strength went down. Okay? So they were improving their muscle strength. We detect antibodies. Now all of a sudden we lose the muscle improvement, but we still have the heart that still is stable. What's this like? Well, it's sort of like, I'm from Detroit, so I think of things in cars, how cars work. Okay? A normal person produces this GAA. It's like a car. It's got the body and the engine. Okay? These patients here, 101 and 102, or 101 and 102, they produce no car, no engine, nothing. Okay? Because the genetic mutation they have does not allow them to make anything. This patient, 103, he makes the car, the body, but no engine. It doesn't work. That's why he's severely affected, because the engine doesn't power the car. But he still produces a little bit that the body says, hey, you know, I've seen this before, and when we infuse it in, it doesn't respond to it as foreign. And so this is why I really want to clarify this, because it seems to correlate with what was presented just now. Now, the good news, the bad news about this, as far as I see it, is that many of you who are juvenile or adult onset patients have probably had a GAA activity level measured in your muscles. And you probably have some activity, but it's very low. If you have some activity, it means you're making that car and your engine is not working so good, but it's working good enough that you can detect some activity, right? In fact, many infantile patients, half of them are in this category where they produce the car but no engine, period, okay? And we call them CRIM positive. In fact, most Oriental patients are probably CRIM positive. We think all the juvenile and adult onset patients are 100% CRIM positive by definition, because if you have some enzyme activity, you have to be producing something. And we hope that what this means is that when finally we get enough enzyme to treat anybody who's CRIM positive, you should not make an antibody to it. That's the theory. I don't know if I'm going to be proven wrong or not, but that's where we're at right now, and that's what we are trying to test. And I'm testing it in animals, and we're testing it in our patient trial. I want to show you our best responding patient. So this is the one that didn't make antibody. Okay? Here's the muscle histology before treatment. And you can see the disrupted architecture of the skeletal muscle. These fibers are being pushed out of the way by these lakes of glycogen that we saw earlier. And this is after treatment. No antibody, skeletal muscle. This is actually the whole muscle. In fact, our pathologist thought we sent a normal person's muscle, because he scanned the whole thing. And this is just not one little section that we honed in on. This is the whole muscle. Okay? Dramatic response. In fact, here he is wearing the obligatory 2001 Duke National Champion T-shirt. He is 22 months at this age, and we've been sending him in to pediatricians' offices, not telling him whether he's got the condition or not, and he's been passing. In fact, I think he's a potential point guard for our basketball team based on this. Here he's about 2 years old, and he just got this urge to dunk basketballs. So I think we need to recruit him eventually. Okay? So similar sort of features as what you saw earlier. And he's pretty cute. So, what are our conclusions from these studies that we published based on what we're seeing right now? Is that we know that this enzyme works. You've heard it from me, you've heard it from Ans. The enzyme works. The companies are convinced it works because we can show that we measure their hearts, they shrink. That's very important. We see enzyme being taken up in muscle. It goes there and it breaks down glycogen. Now, we think that antibodies may affect the response to treatment. Now, we've only got 3 patients. 2 out of 3 show this correlation. I don't know if it's true or not. All we can do is say, this is the data, this is what we see, what we don't see, this is the responses we see, and we go from there. I don't know if it's right or wrong. Yes? It's genetic. Okay? So you can inherit any mutation depending on what type of mutation from one parent and one mutation from the other parent will eventually determine whether you make any enzyme or a little bit, or whether you're CRIM positive or CRIM negative.
I'm talking about the enzymes you were given.
Oh. No, we don't know. So what he's asking is, what's this antibody reacting to? What part of the enzyme? Maybe we can block it, and we're looking at that. But I think it's more so that we're infusing something into the body that the body's never seen before, because that's why you have the disease. You don't make the protein to begin with. So the body, when we infuse it in, it says, this is foreign. We've never seen this. It's an invader. Okay? But in the patients that, like I said, produce this partial protein, they don't seem to see it as foreign, and they don't seem to make this antibody. And then the enzyme seems to work a lot better, not only in the heart now, but then we start seeing these improvements in skeletal muscle. Okay? So we need a lot more studies. Okay? And we think, and I agree, stage of disease earlier, got to be better. Can't hurt. Can't hurt to treat somebody who's still got muscle function versus somebody who doesn't. Those are big questions we don't know yet. How far along are you in the disease that you're maybe no way back? I'm optimistic. I think muscle is highly regenerative. There's muscle stem cells in your bone marrow that I think if you stop the insult in the skeletal muscle, that given time, eventually your body would produce more muscle. I don't know how fast. No one knows how fast because no one's had the opportunity to treat a muscle disease. So, what we're doing now at Duke is we've expanded our trial. We've got more enzyme. And I'll tell you that the enzyme is difficult to make because, like I said, it's got to retain these signaling sequences. It's got to be able to target. It needs to cleave the glycogen. It's not easy to make. And at Duke, we have a protocol in place that as soon as we had enough enzyme to guarantee 1 year of enzyme for a patient, for all the patients in the trial, that's when we started. Because you see, we can't just take someone for 2 weeks and then say, oh, we ran out of enzyme, you know. It worked great for those 2 weeks, but we don't got any more. can't ethically do that. And so you can't start a trial without having enough on the shelf. And I've had a few patients come because they want compassionate use. And I don't have a pile of it sitting in my office. So we're looking at safety. We're still in the safety phase of this enzyme. We need to know if it's safe. And that's what a phase 1 trial is, looking for safety, looking at the doses. Phase 2 is looking to see if it actually does anything. Now, since we're doing it in patients that have Pompe, we have to look and see if it's doing anything. And so that's why it's called a phase 1 / phase 2 trial. Okay? So we're looking at all kinds of functions. And I just want to tell you that, we've enrolled 5 patients more now, infantile, classical, and I can't go into the details because it's an ongoing trial. And I risk FDA staring down on us, if I present data. I will tell you, though, that all 5 patients are showing the same responses in their hearts. In fact, it's faster. Their hearts are shrinking just like we saw before. And so we are continuing to see dramatic responses to this CHO-derived enzyme. Okay? So I'm going to stop there. I just need to point out that there's a lot of people involved. Obviously, Dr. Chen has spearheaded a lot of this from the beginning. He was able to recruit Synpac Pharmaceutical to help us in this effort way long time ago. They really invested money in something that really is a reach, I think. And they were willing enough to do that, and that powered a lot of our early studies. Dr. Kishnani, who's not here as well, she is running this next trial, and I'll tell you, she is running it. I work along with her. But if anyone is more dedicated to getting every patient and baby as fast as Enzyme is available, she is. And I've seen her pull strings in that university to get our IRB approvals in place so that as soon as we were cleared to have Enzyme and a baby hit the door, at that point we didn't have to turn somebody away. Okay? So I can't say enough about her. All the other cardiologists, pulmonologists, genetic counselors, nurses, everybody involved, people in my lab that I'll tell you about a little later. At NIH, for some of our animal work with the knockout mice I haven't talked about just now, Nina Rabin is here. She generously supplied this stuff to us. Just as soon as I ask, she sends it. I wish that were the same for all investigators. National Institute of Neuroscience in Tokyo, Dr. Kikuchi sent us the quails, brought them over, brought the eggs over, talk about going through customs. And we established a colony at Duke. So we have quails, we have mice, we have all kinds of, it's like Noah's Ark over there sometimes. And the support of Genzyme, which has supported our clinical trials as well as a lot of the research I'm doing at my lab on gene therapy that I'll tell you about later. So that's for tomorrow, but I'll stop and I'll answer any questions. Thank you.
Andy, how does the level of enzyme activity in skeletal muscle in your patients who was the most successful compare to the levels of the enzyme activity in skeletal muscle in Ans and the Rotterdam trial?
So we published, so that's Dr. Rabin. She's very nice. She took me out to the Netherlands, actually, when I was out there to Amsterdam. We had a good time there, too. Now, what she's asking is how much enzyme actually got into the muscle. When we published our paper, we measured it. It was a level of 8, I believe, which was just, wait, I'm thinking of the glycogen. Enzyme levels were not as high as what was achieved with the farming trial. The glycogen responses were similar, though. We have similar drops. We brought the glycogen in our one patient down to normal levels. So even though there was probably less activity, glycogen was reduced just as significantly, and you saw that was measured, and we published that number, so it was down.
Andy, just for the record, what dose did you use in your study?
It was 10 milligrams per kilo once a week. Or, I'm sorry, 5 milligrams per kilo twice a week. That's the first trial. So we're looking at lifelong infusion. That's not a cakewalk. And we have seen, you know, sometimes you get fevers, you get shakes. It's not easy for the babies to get this enzyme. It is not easy to be in this trial. And as I told you, some of the babies, we had a response, and then the skeletal muscle response faded. It's a brutal reality that our families are facing, that they had something and it's snatched in some sense. Just like when you are diagnosed with a juvenile onset where you were progressing normally, then all of a sudden symptoms come up and you slowly see things weaken. The progressive diseases, I've seen every disease, or most of them. Progressive diseases are the worst psychologically. They're just difficult to manage, and so we have to deal with that. So I just have to say the families that we have in these trials, they go through a lot. They are putting up their children, they're second-guessing themselves all the time. It's very difficult.
Andy, great presentation, thanks very much. I think a lot of us here amongst the patients are looking at these two presentations we've seen, which are both very heartening news, literally. And it seems to me that there are differences there. And the differences are something that I'd like you both to comment on. Okay, now, one question I have, and maybe this is more for Ans than yourself, was there an antibody response to the patients in the trial with the transgenic enzyme? And if so, did it have the same effects as we've seen in the Duke trial? That seems to me a key question that maybe you can both comment on.
I'd let Ans answer for the farming trial.
Yeah. In the patients there was an antibody response, but also in the good responder and also in the juvenile cases. And to my opinion, as far as we saw, it did not interfere with the uptake of the enzyme in the muscle. As I showed you, we gave the large molecular form to the patients that started, and then we looked inside the muscle whether it ended up, whether it is processed to that lysosomal form. That occurred in all the patients, independent whether they were CRIM positive or CRIM negative, if I go in that direction. So that's and then furthermore, the clinical effect, it is indeed true that 3 of the patients are currently ventilator dependent, but the CRIM negative patients only became ventilator dependent when she was 2 years of age. So, and she's still sitting, she's still making progress. But, yeah.
We measured antibodies. We published that data. Now, there are, I think, scientifically, there are differences in how we're measuring antibodies. So one could say we don't see, we do see, or we see, we don't see. We call this CRIM positive. Maybe it's not if we do a different assay. We're trying to, we backed up our data with DNA mutation analysis that confirms the protein patterns we see. Now, the key is, when I do a lot of our gene therapy work in animals, I'll tell you, trying to develop these ways to come up with treating this with gene therapy. And I get the data, and I go to Dr. Chen, and I say, look, we got this enzyme, it's secreted, and the virus is going in, and blah, blah. And he always asks me, what's the glycogen? What is the glycogen? He just don't show me any, just show me the glycogen, is what he always asks. Because that's the final result. If the enzyme targets, if the enzyme gets in, if the enzyme gets the lysosome, and it can break down glycogen, then the glycogen will go down. So all this other stuff, it basically boils down to glycogen. That's why I asked about glycogen, because that's the final. There's a glycogen storage disease. You get rid of the glycogen storage, you don't have the disease. So how you get there is all these different methods and modes or whatever, but that's the final goal. Yes?
Any thought as to why the CRIM-positive patients did not show regression in their cardiac function, heart muscle problems, while they were concurrently showing problems in their skeletal muscle?
Oh, it's a CRIM-negative patients, yeah.
Right, CRIM. No, this was the CRIM-negative patients.
So like I said, so their hearts, we've converted all of them. Their hearts are treated. They're stable. We've shrunk their hearts down. They maintain stable function. We know we can convert them. Then it comes into this CRIM business as far as we think. But like I said, that's an hypothesis. So in our paper, we talked about this. Why? It turns out that heart has more of those receptors than skeletal muscle. So it can conceivably suck up more enzyme than a similar amount of skeletal muscle. The density of that receptor on that heart muscle makes it that even if there's a little bit of enzyme, it's able to pull it in. That's one thought we had. The other thing is heart is a very active organ. It's pumping, it's got blood flow, it's maximally perfused. Maybe it has access to enzyme more readily being circulated and has more opportunity to pull it up, on top of the fact that it's got these receptors that are a higher level. And it really, if you think about it, it makes sense, right? If you have no enzyme activity, zero, you're an infantile patient. You have heart disease and you succumb to that. If you have a little bit of enzyme, that heart seems to be preserved. But now the skeletal muscle is the real bad component, and you become ventilator dependent or wheelchair bound, because it looks to me like there's a differential response in these tissues. Heart doesn't need as much enzyme as skeletal muscle. And in fact, we've copied this in our gene therapy trial, same response. We can give lower doses of our gene transfer vector, only the heart is corrected and not the skeletal muscle. So it bears out, and it even happens in quails too. I mean, I didn't even know if quails were so diverse, and yet the same sort of mechanisms seem to be in play. Yeah,
Real quickly, I noticed that even with maintaining ventilator dependence, you have some pretty significant physical decreases in heart size. What effects did that have on pulmonary function for these little ones?
Well, so the question is, we had the heart shrink, but yet their pulmonary, what's going on with their pulmonary function? Well, what happened with their pulmonary function is they couldn't keep up, and eventually they required ventilator assistance. Okay? So remember, in order to breathe, there's two things. You got to have good lungs, and then your muscles pump your lungs open and closed. If you have skeletal muscle disease, you can't do that. Okay? So the heart, at the levels we're giving of the enzyme, responds. But then what happens with this antibody may be influencing the access of skeletal muscle, including your breathing muscles and your diaphragm. Diaphragm is a big skeletal muscle. It doesn't get the same amount of enzyme. Now, our one patient, though, that did not make an antibody, and you also saw in the other trial, we've got some patients that they're definitely out on ventilators. In fact, they're out loose, playing basketball and riding bicycles. That's not a ventilator dependent. That's a difference. You see, so skeletal muscles responding in that situation. Arnold.
You did mutation analysis. Did you express these mutations? And
yes,
in what depths did you analyze? And was there any residual activity? Or
there was no RNA transcription
in
by RT-PCR.
In none of them.
None of them, right?
And the one patient you do because you have protein synthesis, so there must have been RNA.
No. In 101 and 102, they were negative, complete nulls. 103, we have obviously some expression.
Right. And did that lead to any activity in the cells, or?
No.
Nothing at all?
Less than 1%.
And did it mature to 76?
I can't tell you that. I don't know off the top of my head.
And the other thing
I showed you in actually his muscle biopsy, we don't detect any 76. We just detect the precursor. So it looks like he's got a maturation problem there. He produces the precursor, but then no mature form.
And your hypothesis then about the crimp status and the antibody response, the idea then is that maybe of the enzyme that you infuse, maybe 95, 98% is neutralized by antibodies, and the 2% is still enough to correct the heart failure?
That's another possibility, is that the body can't make enough antibody to block the enzyme from getting into the heart.
So did you try then to increase the dosage? I mean, if there's some enzyme not blocked, it's just a matter of
yes, we have
a higher dose, and you would rescue.
We're in the process of testing that, yes. I can't tell you, we haven't seen dramatic responses, but we're trying to overwhelm the antibodies by giving more enzyme in the hopes of sopping it up. I think one other point is, I think what you're alluding to is I think one thing that's brought up is, are we sure that our one patient that's responding truly was an infantile patient? That's been a question that's been raised by a few people. I mean, the fact now that he's 2 years old, I think clearly demonstrates that even if some people were suggesting he was one of these crosses, one of these severe juvenile but not quite infantile. And I think the age and the fact that, unfortunately, this family suffered the loss of another child with the exact genetic mutation who succumbed to typical infantile Pompe disease confirms that he truly had the infantile form. And in fact, that muscle biopsy, if that doesn't convince you, I don't know what will. Yes? You have to... yeah. Oh. Okay, so the question is, why are they on ventilators? Is it because the muscles can't contract and move those lungs? Or is it the lungs actually have a problem with getting oxygen into the body? I think the lungs are normal in these kids. It's their muscles and the rib cage around it can't open and close appropriately to get air in and out. So it's clearly not intrinsic lung disease like, let's say, a cystic fibrosis kid, who their disease is because their lungs are not functioning, their muscles are just fine. In this instance, you have a myopathy. And so I've been talking with some of the families that when it comes to ventilator support, think about it as a muscle weakness disease, not an intrinsic lung disease. And that will change the pulmonologist's view about how those vent settings should be. You may think about other forms or even extrinsic sort of compression of the chest rather than trying to do it like a conventional lung failure patient whose underlying process is the lung just can't work. Clearly distinct. Yes?
Have you seen any anecdotal evidence that deleterious mutations or DNA induce disease?
So your question is, induced by what?
Induced by whatever. Other than here.
Oh. Well, the question is, can a GAA mutation in a DNA be induced? For example, you didn't inherit it. It's a recessive condition, right? That means in a recessive condition, everybody inherits two sets of genes, one from their mom, one from the dad. In a recessive condition, both copies of a gene have a mutation in them and you get the disease. Okay? Many times if you inherit, so if you have two copies of a disease, it means you got one from your mom and one from your dad. Many times, though, you go back and you look at the family, and the mom has the disease, or the dad has the disease, you find it in his blood, but the mom you don't find it in their blood, and you're like, well, what's going on? Well, it turns out that the mutation probably occurred in the mom's eggs, in the oocytes, in the ovary. Okay? So it happened at some time during the ovary developed that a few of the eggs had the mutation, but not the whole body. Okay? That's normal. Or it could happen in the sperm. So you check the man's blood, you don't see the mutation, but you look in the sperm, and a certain proportion of them have the mutation. That's where the first event occurred. So that's how it can appear to be sporadic, but in fact, it happened at that point, and it was when the egg and sperm combined that then you end up with an individual that has both genes mutated. Okay? So that happens in other diseases too.
Do you ever see any anecdotal evidence that it can be?
No, I don't think so. I don't think there's any reported cases where, let's say you inherited only one copy of the gene. You should be fine, right? Typically, because it's a recessive condition. And let's say something happened to you that now your other copy got mutated, let's say by radiation or whatever, then theoretically, but your whole body would have to be affected. Every muscle would have to get that mutation. That's not been reported.
Andy, I think maybe Harold's asking a question. Can you stimulate the mutant gene to produce more active protein?
Oh.
In some way, by adding a component that may turn the mutant protein.
Back?
No, the mutant protein. If you could put something into the patient that would activate the mutant protein so that it gave more activity.
There are ways to think about that. That's difficult biochemistry. I mean, right now, and it's sophisticated. I mean, we're kind of taking a hammer to this. You're missing enzyme, we must give you enzyme. You have a DNA defect, we give you DNA. There's other levels of complexity on top of this. Can we modulate this protein, maybe somehow give you a drug to make this enzyme fold differently so now it functions instead of a 2% level, maybe a 5 or 8% level, and move you from a severe juvenile maybe up to an adult. Those are things that certainly are on the drawing board. In fact, the stuff from Novozyme is playing on this. How can we make this enzyme a little better to maybe improve its ability to get in? I mean, you'll hear about that. But there's all kinds of these. Everybody's got their own little devious little plans on how they're going to make this work a little better. And I certainly have some too, but that's one possibility.
Andy, I'm afraid, I think maybe Randall, do you want to call it to an end, this particular session?
If I don't want to hog, I could step down and...
Okay.
Actually, Johan here had his hand up for a while. I have two questions, one to Andy, one to Ans. Andy, in your hypothesis, with antibodies blocking the amount of enzyme going to the muscle, you would expect to have less enzyme in the muscle from patients 1 and 2 compared to patient 3. Is that correct? And how, and what a percentage compared to normal do you see in the muscle?
We did not see that. Did not see that.
Does that not fit your hypothesis?
But we did see differences in the glycogen. So remember, when you take this hunk of muscle out, what are you actually taking out? You're taking blood vessels, you're taking muscle, you're taking a lot of things. If there's some enzyme in there, it may not even be in the lysosome. It may just be hanging around, sticking to something. So activity levels, again, really are not the final answer anyway. Glycogen. Is the glycogen down? That's the point, I think, is that. So activity levels, whatever they are, if they're sky high, but the glycogen's not down, that's concerning too. Same sort of thing. We did not, in our measured levels, I don't think we saw very significant differences. They weren't very high to begin with, but certainly the end result, the glycogen was corrected in the one patient.
What was the percentage of normal of enzyme activity that you
I don't have the number off the top of my head. I've got it. Where's my briefcase? I'll give you the number. But it's only, I know we're not approaching 100%, 8%, something like that. So we're sort of like a juvenile, infantile sort of individual. Certainly not 100%.
A question to Ans. When you have such a high enzyme activity levels in your muscle, why does that enzyme not break down the glycogen?
That's an intriguing question. If you measure the glycogen in the laboratory, you saw what happens. It levels out, it stabilizes in 3 patients, and it goes down in 1. But what we also did, we looked at positive material. And what's positive? You use a certain dye to stain the glycogen. And then we did a kind of scoring mechanism. We did that blindly in the laboratory. So we're sitting with a large number of people around a microscope. You have a central microscope, and then we made a score. We did not know which biopsy it was, whether it was the first or the last. And each time we scored the last biopsy less in intensity of pus staining with respect to the first. But what I also told you is that when the so there seems to be an effect on the lysosomal pool of glycogen. We do not have an explanation why you don't see it in the total glycogen amount. And what we saw is that, it corresponds very well with how the muscle acts in the patient. If you have a good function in the patient, then you see the lowest amounts. Is that an answer?
Is there any evidence for an inhibitor?
For an inhibitor?
Inhibition.
Can you explain? Do you mean in the cell, or
you get enzyme in the cell?
Yeah.
It doesn't do its job. Is it inhibited?
No, I think it has to do with the function of the muscle.
And is it sitting in the right cells, in the right portion of the right cells?
I think so. I think so. But
well, again, a question for you both. When you hear so much positive results, it's hard to inject maybe a kind of sour note, but I feel I must. I have a serious concern here, because here we have these 2 trials, which have, are significant advances, and yet there seems to be differences between them. And I, as a lay person, find it difficult to say whether those differences are significant or not. Okay, so on the one hand, we have results here that show with one type of enzyme, there's antibody production, and that has a significant effect on the treatment. Okay. On the other hand, from the Rotterdam trial, we're hearing that there was antibody production, but it did not have the same effect. Now, I appreciate there's different methods being used and so on, but this is a fairly fundamental matter that must be addressed. And what I'd like to know is, how are you guys going to address it? How are you going to look at these two trials and work out whether they are in fact actually telling us the same thing or whether there is a difference? And maybe this is a question for the people from Novozyme and Genzyme as well.
I think it is, but I will say that in our trial that we've just started, we are trying to attempt to clarify this. We're trying to deduce the CRIM status of our patients, look at their antibodies, what type of antibodies. Not all antibodies are the same. We may be measuring one type of antibody, you may be measuring another. And I don't see the discrepancy you see. I see two enzymes that work, that seem to work, that in some patients it works really good, in other patients we certainly shrink everybody's heart. It works. And that stabilizes the theory that if you infuse this enzyme into somebody, it will get into their muscles one way or the other. Now, the difference is there's just not enough patients. I mean, Ans said it. This is a very limited trial. We don't have the luxury of doing like a blood pressure trial. You got your new blood pressure medicine. Let's try 10,000 people with blood pressure and see a 2-point drop in systolic blood pressure. There's not enough enzyme to do the really sophisticated kind of clinical trials that are going to be needed to delineate this. All we can do is move forward as we get enzyme available, try and condense down as much info as we have, try and make every piece of data count, and then hopefully we move forward. And unfortunately, those are the decisions that are left up to the companies to make about other things besides, they have to look at the efficacy, which is how good it works, they have to look at the side effects, and then they have to look at all kinds of just practical capitalist type issues that boil this all down.
Ans?
I think you more or less gave an explanation, and that's okay with me.
I tell you, I see more agreement here. I just don't see that glaring differences. I mean, you can get into technical things, but I walk away from this meeting saying we're seeing the same results. We are seeing responses. I'm telling you that other muscle diseases like muscular dystrophy wish that they had this sort of mechanism to take part of. Duchenne muscular dystrophy, there's no way something like this is ever going to work for them. You have to get the gene into every muscle cell. Here we have a biological method of sneaking this enzyme in through the blood. To prove that is tremendous. And we proved it two different ways. Two different groups did it two different ways, and we proved it. There's no one outside of this room that can say, this therapy is never going to work for anybody.
Could I suggest that perhaps one of the differences between the two trials is the enzyme itself that you're using, and they have different carbohydrates and energetic sites?
Certainly.
It's just a suggestion.
Oh, I agree. But like I said, this is a phase 1-2 trial. In both instances, we're looking at safety, and then if we can get any efficacy data out of it, we'll take it. But it's not meant to be the be-all to end-all trial. That's why it's called a phase 1, phase 2. We're just looking at, can we infuse it safely, repeatedly? And then if we get a result, well, that's gravy. That's frosting on the cake, and we both got it.
As I understand it, they're going to start a totally different trial, and they're not going to use the data from these two trials to compare or make comparisons or to gather which one would be the most effective. Now, in the Rotterdam trial, they did not have where the muscle function of these babies deteriorated to pre-infusion levels, but you did. So it seems to me that we need to gather the 3 different methods and see which one would be most effective. Now, I, as a parent, want my child to improve, not just for heart disease, but I want that child to have a life where they can carry on and raise their head, move their arms, be able to eat on their own, that kind of thing. So I think this is what we need to all concentrate on, and to pick out the therapy that is going to be the most effective with the most amount of people in the shortest amount of time.
I agree 100%. I do think, though, that based on the published data and what we've heard today, that I think that the enzymes are working similarly. We see the same response. We see the kids playing basketball, for God's sake. We scored him out. He scores normal on those Ames tests. We see a response just as effective.
When we first started this back in 1996, I was very much a novice, and I made a comment at one of our scientific conferences that, well, as long as we get the enzyme is enzyme. And Dr. Michelle Hirschhorn stood up and she said, enzyme is not enzyme. And I want you all to know that. There must be differences in which I have not knowledge.
I'm not. Certainly, there's difference. I mean, one's made from a rabbit, one's made from a hamster ovary cell. There's all kinds of.
We have enzymes. We all have the same.
I wish, but like I said, this is a molecule that has multiple functions. It has to target. It has to get in. It has to break. All those things cannot be degraded on the way to actually injecting it into a patient.
You're not making the same kind of enzymes that a normal person makes?
No.
So your enzyme is not identical to the normal person?
It's close at the protein level, but actually these proteins actually have little kind of embellishments on them called carbohydrates that I'm not an expert. Yeah. Yes. 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 They do differ at that level. But like I said, Dr. Chen said, look at the glycogen.
Perhaps it's important to remember that the human enzyme from humans was used in 1972. It doesn't work as a therapeutic drug.
It's complicated. I mean, I try and simplify this down, and in the process of doing that, I'm biasing it, okay, to our point of view, and I'll freely admit that because I have a view based on what I see, and then other people, you have the same data, you can distill it down to what you want to present. All I'm trying to do is just educate and see it from my point of view, and I'm an optimist. I see two enzymes proven by two different groups, done totally different places, and they get similar type of results. That's huge. There's all kinds of things being done to people out there that are not based in any kind of fact as solid as that.
Dr. Amalfitano.
Yes,
so
Dr.
good to see you again after 10 years, to see you and to hear you speak. As a generalist, I have a two-part, very basic question that's totally off of what's being discussed right now. The first part of the question is, what exactly is harming the muscle cell? Is it the crowding due to the accumulation of the glycogen, or what? Is it poisons from the lysosomes, or is it the lack of glucose? That's number one. The second part of my question is, why in the same patient, if it's a genetic disorder, are some muscles affected more than others?
Whoa. Don't know and don't know. Dr. Reusser probably knows more than I do. It's bad to have abnormal amounts of glycogen in there. Maybe it's breaking the lysosomes open and they're spilling their guts into the muscle and it can't work as well as it should. That's one theory we're looking at. I think Dr. Reusser is the one. If you have any? Yeah? We're kind of dragging on?
I think it's probably a good time to stop this session at the moment and have a break before the next session, because I'm sure this cycle of questions will continue. Said I'm mother for the rest of this session, and the first speaker, Dr. Klinge, is going to tell us about enzyme replacement therapy in infantile Pompe disease from Essen, Germany. Thank you.
Can you hear me all right? Is that all right? In the back, can you hear me well? Very good, okay. First of all, I want to thank you very much for inviting me to this beautiful place. I've really already enjoyed being here in Texas, and it's so nice. And it's so nice meeting people of you, and it's been so motivating to feel the spirit that is in this group, and I'm sure I will take some of this home back to Germany with me. I'm going to report on enzyme replacement therapy in infantile Pompe disease in Essen, Germany, as it's taking place in our department of pediatric neurology. First, let me shortly introduce our team, since only the team is what makes this clinical trial possible. We have Thomas Bosbach. He's our physiotherapist, not only involved into physiotherapy, but also in the assessment of the development scales. We have Bärbel Leindecker, our dietitian, the nursing staff with a huge impact on the functioning of a clinical trial like this. Volker Straub, involved into administrative work. Thomas Freud as head of department and principal investigator. And I'm involved in the daily work on the ward with the patients and their parents. I will try to focus on this aspect in my talk. Currently, we have 4 patients under enzyme replacement therapy in our department in Essen, Germany. 2 patients are under medication derived from milk of transgenic rabbits. We administer a dosage of 40 milligrams per kilogram once weekly. And 2 patients are under medication produced through the genetically modified Chinese hamster ovary cells in a dosage of 10 milligram per kilogram once weekly. Patient 1, she's a girl from Holland, actually. She's been enrolled in the study ever since the age of 3 months. She's 21 months old by now, and she's admitted for therapy once weekly. She fairly much lives the normal life of a young girl. They live about 3 hours away from our hospital, which in Texan relations is not really far, as I understand. And so therapy day for her looks like getting up in the morning. They leave home, arrive in the hospital around noon, and after all of the nurses and sometimes even the doctors have been hugged, we start therapy lasting until about early evening. She spends the night, goes home next morning. Parents always like to use the afternoons to go shopping in Essen. She is on no more concomitant medication, no more cardioprotective medication, and that's basically due to the fact that the cardiac function is in the lower norm by now. Her weight is below the normal range. She does not have any hearing problems, and she does not have any additional oxygen supply. What about her development? She's progressing in her development. She has been able to sit unsupported ever since the age of 1 year. She's using her arms. She's doing that well and quite effectively. Actually, just the other day, in a very short, unwatched moment, she grasped one of the infusion bottles and lost interest quite quickly and threw it over her shoulder, which of course was kind of bad for the medication, but was a very impressive demonstration of her muscle strength as it is now. She can only eat soft foods. She has problems biting and swallowing solid foods. She's talking in 2-word sentences at the moment. Her developmental mental age ranges around 16 months, being 21 months old now, and her developmental motor age around 6 months at the moment. And she's a very well-balanced character. She's quite happy in herself as a child. What about patient 2? He's a boy from the United Kingdom, Furters. He's been enrolled in the study ever since the age of 6 months, 17 months old now, and he's been an inpatient ever since the beginning of therapy. His clinical state has not allowed us yet to discharge him. And this is mainly because of the fact that he's suffering from frequent pneumonia, mainly